Okay, so this is our genetics project on biocomputational analysis of protein sequence of sickle cell anemia by Adia Burley.
Jalen Carter.
Aaron Seattle.
Shazne Kalu.
and Robert Price. Sickle cell anemia is a genetically inherited disease passed down to offspring from two parents who both carry the sickle cell trait. This abnormality causes various health effects in individuals who suffer from the disease. The blood cells are sickle shaped instead of spherical, which decreases their affinity for oxygen. As a result, a person can suffer from various complications throughout life, such as anemia, bone and joint pain, joint swelling, recurrent infections, etc. Biocomputational analysis examines the protein responsible for the change in blood cells. In this research, various protein sequences analysis programs are used to study 9 different sickle cell proteins.
This study focused on analyzing the protein sequence of sickle cell anemia using various biocomputational tools. Results showed that the protein is highly hydrophobic, which affects its structure and behavior in the body. It was also found to be thermally stable across a wide range of temperatures, including tools including SLPMA and Swiss model were used to examine the protein's physical and structural properties, revealing a high presence of alpha helices and random coils. These insights provide a deeper understanding of the disease and offer a foundation for drug development and future therapeutic strategies.
So our research question and main findings were that the research aimed to understand how the structure of sickle cell protein contributes to the disease effects in the body. A specific mutation causes a replacement of a hydrophilic amino acid with a hydrophobic one, leading to the sickling shape of red blood cells. The analysis highlighted key conserved regions, structural stability, and hydrophobicity, of all which play a role in the disease progression. These findings can support drug design by identifying potential targets within the protein for treatment or intervention.
Now, when it comes to background, this study focuses on biocomputational analysis of sickle cell anemia, a genetically inherited blood disorder in which red blood cells adopt an abnormal crescent or sickle shape rather than the normal round shape. This distortion reduces the cell's ability to carry oxygen efficiently, often leading to severe complications throughout the body. The abnormal shape arises due to a single amino acid substitution in the beta chain of the hemoglobin protein, where hydrophilic glutamate residue is replaced by hydrophobic valine residue at the sixth position. The substitution causes hemoglobin molecules to aggregate, distorting red blood cells and impairing their function. Sickle cell disease affects approximately 2 million people worldwide, and over 300 million individuals carry the sickle cell trait. While individuals with the trait often live normal lives, those with the disease may experience symptoms such as chronic anemia, bone and joint pain, joint swelling, and osteomyelitis, necrosis, and splenic complications.
To add on to the background, despite its global prevalence, sickle cell anemia is often less recognized compared to other diseases like malaria. However, it often remains one of the most common and disruptive genetic disorders. Understanding its molecular basis through computational biology can reveal crucial insights into the structure and function of the affected proteins. Tools such as ProPartum, SOPMA, Swiss model, and GeneD allow for a comprehensive analysis of protein structure, hydrophobicity, thermal stability, and sequence conservation. This study utilizes a range of bioinformatic tools to analyze the protein sequence associated with sickle cell anemia, aiming to identify their physiochemical properties and structural characteristics. These insights are vital in supporting future research and therapeutic management of the disease.
For our context and significance of our study, we said that like sickle cell disease was a family of blood disorders, including like sickle beta thalassemia and cell anemia. A preponderance of red blood cells that become hardened and brittle and sickle shaped when they release oxygen. Sickle cell disease is caused by an amino acid replacement in the beta chain of the human hemoglobin with glutamate replaced with valine. Um, Valine is a nonpolar and hydrophobic and tends to shrink in the middle of protein. The deformed protein forms bundles and distort the red blood cells that carries them, resulting in the characteristic shape of a sickle.
The purpose of the study is to explore sickle cell anemia through biocomputational analysis in order to characterize its protein sequence and determine its physiochemical and biochemical properties within the living organism. Gaining a deeper understanding of these properties can provide valuable insight into the structure and function of the affected proteins, which may play a significant role in the progression and management of the disease.
So to be proven, um, to be proven is to perform biocomputational analysis of sickle cell anemia in order to characterize the sequence and to determine its physiochemical characteristics in order to understand progression and management of the disease.
Now, some of the main results that we found highlights key data, findings, and figures. The result of this research was better understanding of sickle cell disease, basically, as you can see in the graphs, and the protein responsible for the change in the blood cells. By using bioinformatic tools to take a closer look at the sickle cell protein, researchers basically were able to determine its structure, sequence amino acid frequencies, and predicted RNA structure. The bioinformatics methods proved to be useful for identifying novel drug targets for the relevant protein structure. This research may be used in broad screening of inhibitors of the protein. So there's some description as you saw in the pics. Figure 5 shows 3D predicted 3D models of the sickle cell hemoglobin protein built using the Swiss model tool. These models help visualize the protein's overall structure and folding patterns, which are important for understanding how the mutation affects the function of it. Figure 6 goes a step further by showing basically the protein with a ligand bound to it, highlighting the potential drug binding sites. This is useful for identifying how treatments might interact with the protein and for guiding drug design.
For the interpretation of the results, the study used bioinformatic tools to better understand the proteins involved in sickle cell anemia. The results show that these proteins are mostly hydrophobic and thermally stable, which means they can hold their structure across different temperatures. Most of the protein structures were made up of alpha helices and coils, which makes sense given the type of amino acids found. The conserved regions found in the protein sequences could be important for how the disease works and might be useful targeted for future drugs. The RNA structure also showed good stability based on free energy calculation, and the 3D protein models helped show possible binding sites that could be useful for treatments. In terms of the study limitations, one major limitation found is that everything was done using computer programs and not any real lab work or real world testing was done to back up the models. Also, the study only looked at 9 protein sequences, so the results might not apply to all cases of sickle cell anemia. Finally, since this wasn't linked to any actual patient data, we can't say for sure how these findings relate to symptoms or outcomes in people.
So, potential implications, even with its limits, this study shows how useful bioinformatics can be. It's a fast and low-cost way to study diseases like sickle cell anemia and could help researchers find new drug targets. The conserved regions and structural details might be used to create better treatments or even new diagnostic tools. These kinds of studies also help us better understand how the disease works at the molecular level. For as far as future research goes, it will be important to test these models in the lab to see if they match what's really happening in the body. Researchers could also look at a bigger variety of protein sequences, including ones from different people, to see how much the findings hold up. It might also be useful to run drug simulations to find out what molecules could bind to these proteins, potentially stop the sickling process. Adding in patient data could also make the research more personalized and impactful. For context, this study was done in the early 2010s when bioinformatics was becoming a powerful tool for studying diseases without needing a full lab setup. Around that time, access to genetic data and online tools like the Swiss model and Parum were expanding, making studies like this more doable. Also, there was growing global attention on these diseases like sickle cell anemia, especially in areas with limited healthcare resources. So using computer models was a smart way to keep moving the science forward without relying on expensive lab work. Thank you.